All right, this is for your Chapter 7 test. Make sure you have your study guide handy. If you don't have your study guide, that's okay, because I've copied and pasted the questions into this presentation. So let's go ahead and go through your study guide. Make sure you get an A on this upcoming test. So question 1, know the 3 parts of the cell theory. So there are 3 parts, and you have to know all 3 parts. And the first part is, the smallest part of a living thing is a cell. If it's smaller than a cell, we don't consider it alive. So those organelles that are inside the cell, we don't consider them alive, because cell theory states the smallest part of a living thing is a cell. So anything smaller, we don't consider it living. All cells come from existing cells or pre-existing cells, as we said in class. And the third part is all living things are made up of 1 or more cells. It does not have to be this word for word. As long as you're getting the general concept of these 3 things, you'll be fine. It doesn't have to be word for word, and you can put it in any order you want. All right, let's move on. Number 2. So know the different types of microscopes and how they are used. So we have a compound light microscope, and that magnifies things using an objective lens that eyepiece you look through, and lights, which is why it's called a compound light microscope. Compound light microscopes can magnify up to about 1,000 times. After that, the image becomes blurry. If you want to zoom in further, you would use an electron microscope, and there are a couple different kinds. One is a transmission electron microscope, and that is a 2D image. The specimen has to be cut very thin. False coloring is added so scientists can see the image. Electrons don't come in color, and I'm just getting email after email after email here. Electrons don't come in color, so that's why it's a black and white image. Scanning electron microscope will give you a 3D image. Just all kinds of emails here. That's, I wasn't getting any emails before this video started. Now we're just getting it all over the place. That's just great. But anyway, scanning will give you a 3D image of the specimen. They don't have to be cut very thin. In transmission, you're going to get a 2D image, and that's because the specimen has been cut very thin. And the reason electron microscopes are black and white is because electrons don't come in color. All right, moving on. So compare, contrast prokaryotes and eukaryotes, along with knowing how to classify each one. What's an example of a prokaryote? Okay, so eukaryotes, prokaryotes. When you hear these words, you should be thinking one word: nucleus. One of these have a nucleus, and one of them do not. Eukaryotes have a nucleus. They contain a nucleus. And in some examples that we talked about in class, animals are eukaryotic. Fungi, like mushrooms, they are eukaryotic. Plants, they are eukaryotic. They also have a cell wall and a membrane. Animals do not have a cell wall. Prokaryotes are cells that do not contain a nucleus, and we talked about bacteria, and they have a cell wall and cell membrane as well, but they have a cell wall. So that's it for that one. Just remember that eukaryotes have a nucleus, because the EU in eukaryote means true nucleus. Prokaryotes, the PRO means before there was a nucleus. All right, so know the organelles of an animal cell versus the plant cell. You can see the diagram. I copied and pasted it from the bottom of your sheet here. You need to know that ribosomes make proteins. You can find ribosomes on the rough ER. That is the rough endoplasmic reticulum. That deals with making proteins. We'll get to that in a little bit later. For the ER, or the endoplasmic reticulum, we just went over rough. There's also the smooth ER, which removes toxins from the body. We have the mitochondria, which breaks down food particles to supply energy for the cell. We have the nucleus, which is like the control center. It houses DNA, chromatin. Inside the nucleus, you have something a little smaller called the nucleolus, where ribosomes originate from. We have the plasma membrane, also known as the cell membrane. It regulates what enters and exits the cell. We have the Golgi apparatus. This relates to the rough ER, because the rough ER makes proteins, starts manufacturing them. They are sent then to the Golgi apparatus, and the Golgi apparatus is going to modify, sort, and package the proteins to be used either outside the cell or inside the cell. This open space here that is unlabeled, we call this the cytoplasm. Here we have the centriole that is used in animal cell reproduction. We'll talk about that next unit. All right, swinging over here, we have the giant central vacuole, which is like a storage center, like the UPS warehouse. We have chloroplast, which is only found in an animal cell used for photosynthesis. Here's that cell wall. On the inside of a cell wall, you have the cell or plasma membrane, mitochondria, which we just talked about, everything that we just talked about. But notice in a plant cell, it's going to have a cell wall, chloroplast, and a giant central vacuole. And we'll go more in depth in these in just a minute. So here we have all these right here. We just said all this. So go ahead and pause the video, make sure you have all these, and make sure you're ready. Okay, moving on. One thing we forgot to mention that is not labeled on here is the cytoskeleton. And just like your skeleton, it gives us shape and structure. Cyto means cell. So we're talking about the cell skeleton, which gives the cell its structure and its shape. All right, moving on. Number 6. So what is in a plant cell that is responsible for gathering energy that's not found in animal cells? Well, in plant cells, you're going to find chloroplast, and that's used in photosynthesis. In animal cells, you have mitochondria, which breaks down the food. Plants have mitochondria too. I was just throwing out animal cells have mitochondria because you're going to be seeing that in the next unit. And really what you need to know for this is that plant cells have chloroplast that's used in gathering energy that is not found in animal cells. All right, moving on. No, diffusion, facilitated diffusion, and osmosis. Okay, so 3 big words here. Very, very easy to understand if you study. So the first one, diffusion. Particles collide randomly, and particles move from an area of high concentration, where there's a lot of it, to an area of low concentration where there is none of it. In the podcast that we did, we had bacon and eggs as the example. When you're standing in the kitchen, the aroma is very strong. It's going to collide randomly with the particles, the air particles, and it's going to move away from high concentration to low concentration. You probably have experienced this with those people in the classroom who feel like they have to spray something. They spray something and it makes its way across the classroom. That is an example of diffusion. It's moving from where there is a lot of it to where there is none of it. It happens randomly and it collides with the air particles around it. Now there's facilitated diffusion. It works just like diffusion except for it uses protein pumps or protein channels. If you see the word protein pump or protein channel, it is facilitated diffusion. Now, osmosis is the same thing as diffusion, but there's one key word, and that is water. If you hear anything about water moving, chances are the answer is going to have something to do with osmosis. So it's water entering the cell through a selectively permeable membrane, and osmosis is the diffusion of water. It's from an area of high concentration to an area of low concentration. And that's why it falls under this question. Please know that none of these require energy. And because they do not require energy or ATP, they are known what is called passive transport. That means no energy is required. All right, moving on, halfway through number 8. What happens when the concentration of molecules on both sides of a cell are equal? Well, the first thing we want to do is know what this is called. And this is called dynamic equilibrium. And that's when the concentration on the inside is equivalent to the concentration on the outside. That could be of a cell, it could be in a solution, could be in a box, whatever. The important thing, though, it says what happens when it's reached dynamic equilibrium. Well, the particles don't stop moving. They are still moving and colliding. They're moving in and out at equal rates. So they're still moving, they're still colliding. They don't just stop. They're still bouncing into one another. So even though something appears still and it's not moving, it actually is moving. Those particles are bouncing all around still, even when the dynamic equilibrium is reached. Dynamic equilibrium means the outside and inside are equal. It does not mean stop. It would say stop if those things would stop, but it doesn't. It says dynamic equilibrium. All right, number 9, no osmotic pressure, passive transport, active transport. Osmotic pressure has the word osmosis. You should immediately be thinking water. We have passive transport. We already went over that. That means no energy. And we have this word active transport. So we'll go over that real easy. So osmotic pressure is the amount of water entering a cell. Now, animal cells do not have a cell wall, so too much water can cause the cell to swell and burst. It is passive transport, which means no energy. Then we have active transport, which uses energy. We could also say ATP. Endocytosis and exocytosis are examples of active transport. All right, number 10. So we talked about protein pumps and protein channels earlier. So it says, if a glucose molecule is moving through a protein pump, what type of transport is it? So this is a protein pump. I have my glucose molecules. I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Over here I have 1, 2, 3, 4, 5, 6, 7, 8. And we see it's moving from an area of high concentration where there's a lot of it through the protein pump to where there is less of it, high to low. The reason it's facilitated diffusion is because these glucose molecules are moving through the protein pump or protein channel. So when you see something like that, it's automatically facilitated diffusion. That's the only difference. It's the same thing as diffusion, except for it's moving through a protein pump or protein channel. Please keep in mind, this is passive transport. It does not require energy or ATP. All right, number 11. What's the term used to describe the function of the body to keep everything internally stable? And that is simply homeostasis. Homeo meaning the same, stasis meaning state, trying to keep the same state in the body. An example of this would be your body temperature staying at 98.6 degrees. All right, there is no number 12. That was a mistake on my part. There was number 12 at one point. I thought the question was too easy, so of course I had to take it off. Microsoft Word did not update the numbers, so no worries there. Number 13, is the nucleus in the nucleolus, or is the nucleolus in the nucleus? Well, the nucleolus is inside the nucleus, so E, right here, we have the nucleus. D is the nucleolus. A, here we have the rough endoplasmic reticulum. You gotta look close to see the dots on it, and I will tell you on your test what has dots and what does not. The smooth endoplasmic reticulum is right over here. Here we have the mitochondria. Here we have the Golgi apparatus. B is not pointing to anything. It's pointing to the open space. The open space is the cytoplasm. Let's do I. I is the outer membrane here, or the outer section here. Is it a cell wall or cell membrane? How do I know? Well, I know this is a round shape, so I know automatically it is an animal cell. I also know it's a eukaryotic cell because it has a nucleus. But I is the cell membrane. H is pointing to these little dots, and these little dots are the ribosomes, and they are used for making proteins. So make sure you know all that. Let's keep moving. 14. What does a cell take in water and food through? What does a cell eliminate waste and food through? Well, the cell's selectively permeable membrane. Selectively permeable or semi-permeable means it allows some things in and some things out. If we just said it was permeable, it would allow all things in and all things out. But it's selective about what it allows in and what it allows to leave the cell. All right, number 15, 2 more. So know the order of simplest to most complex levels of organization of a multicellular organism are cells. And if you take a bunch of cells together, they make up tissue. If you take a bunch of tissues and put them together, they'll make up an organ. When you take a bunch of organs together, they'll make up an organ system. So a bunch of cells working together make up tissues. A bunch of tissues working together make up an organ. A bunch of organs working together make up an organ system. So make sure you pay close attention to that. And number 16, endocytosis versus exocytosis. You should immediately be thinking active transport, which means it requires energy. So endocytosis, endo, think in, going into the cell. It is things entering the cell, it uses energy. ATP is a fancy way of saying energy. Exocytosis, things exiting a cell, the exo, giving it away there, also uses energy. Now, a lot of times on questions, they may ask you to compare and contrast, meaning what is similar and what is different. Everybody gets the different part, endo going in, exo going out. They do have something in common. What they have in common is they both require energy. They are both active transport. Active transport means they require energy. They require ATP. All right, so that's going to be it for your test review. Kahoot has also been sent out. Make sure you play that. is no longer live starting Friday at 7 a.m. It'll go down at that point, but you'll be taking your test by then, so it will not matter. If you have any questions, please don't hesitate to contact me. Leave a comment, and we'll see you guys in class.